Hello everyone, hello my dear students, myself Ashish Dubey and we are going to discuss today a very special topic, that is the nerve impulse. See, the nerve impulse is very much popular in the chapter of nervous system, and the conduction of nerve impulse and transmission of nerve impulse are two very important topics that are taught in very detail in this chapter. So let us discuss about this topic, and here we go with the nerve impulse. See, the nerve impulse is generated, then conducted, and then transmitted on the length of one neuron and ultimately to another neuron. So we are going to discuss this concept in a gradual way, in a stepwise way. So I hope you all are very familiar with this structure that we are having here. That is the neuron, and we are having the dendrites, these branching like processes, these one, and then the cyton, the cell body, and this the longest dendrite, that is the axon, ultimately ending in button like structure that is known as the synaptic terminals or synaptic nodes. Okay, so what is a nerve impulse? First of all, let us discuss this. See, we can say that nerve impulse is the information flow from source to processing center. Source to processing center, there is flow of information. Okay, this flow of information is known as the nerve impulse, okay, on the length of axon. So we can say that source, what is the source? Source are the receptors, the sensory nerves. Okay, so I am going to write over here receptor. Okay, so source means the location where the nerve impulse is being generated. What is the nerve impulse? The information flow. Okay, the information that is flowing along with the length of axon. Okay, that information flow is known as the nerve impulse. Okay, the receptor is the cause of generation. Receptor is the cause of generation of nerve impulse along this axon. And what is the cause of generation of this nerve impulse in the receptor itself? We are going to find out very soon in the topic generation of nerve impulse. Okay, so stay connected. First of all, what is a nerve impulse? Nerve impulse is the flowing information from source to the processing center, that is our skin, we can say. Okay, let us discuss this thing. See, the generation of nerve impulse. First of all, we should be familiar with this term, stimuli. Stimuli, it is plural, it is a plural term. Okay, we can say that any external or internal environmental change in the body is known as what? Stimuli. Okay, our body is very sensitive to any type of external or internal environmental change. External environmental change, it means the wind, the light, okay, the humidity. Okay, internal environmental change means the pressure, the partial pressure of oxygen, carbon dioxide. Is there any injury in the body? Okay, the internal temperature, the osmolarity of the body. Okay, all these external and internal environmental changes are considered as stimulus. And stimulus is having the plural as stimuli. So stimulus is required for generation of nerve impulse. That information that will be flowing along the length of axon needs to be generated first now. So these stimuli act as the information. Okay, and from this phase ahead, there will be nerve impulse. So how the stimulus is being converted into nerve impulse? Because of the receptors. As we discussed just a moment ago, that what are receptors? Receptors are nothing but the dendrites. They are dendrites. Okay, so dendrites are capable of receiving these different kind of external, internal, environmental change and causing the nerve impulse to be generated. Next thing is the conduction. So what do we mean by conduction? In the previous slide, you can see here, once the information is generated in the dendrites, then it will flow in the cyton and then along the length of axon up to the synaptic terminals. Synaptic terminals or synaptic nobes. Okay, synaptic nobes. And from here, it will be transmitted to another neuron. So conduction means what? Conduction means what? The information or the nerve impulse is being propagated. That is very suitable word here. It is being propagated along the length of axon, the nerve impulse. Okay, so that is known as the conduction, and ultimately when it will be transferred to another neuron, that will be considered as the transmission of nerve impulse. Okay, so what is the conduction? Conduction is the propagation of nerve impulse along the length of axon fiber or nerve fiber or in a nerve. Okay, so this is the generation, this is the conduction, and what is the transmission? Transmission is the transfer of this nerve impulse from one neuron to another. Okay, when one synaptic nobes are ending on the dendrites or on the cyton or on the axon of another neuron. Maybe there may be a dendrite or the cyton or the axon itself. Never the synaptic node is in communication with another synaptic node. Okay, it doesn't happen. It doesn't exist in our body. Okay, so next thing, what are the stages of this conduction of nerve impulse? Okay, so first of all, we have to consider three stages in our mind. That one stage is there where no impulse is there. Okay, the membrane, or we can say the neuron is resting. Neuron is resting, it means there is no stimulus. Okay, it means we can say there is no change in temperature, there is no change in pressure, there is no touch, there is no light, we can say, or there is no change in the light intensity. Okay, like that. So that will be considered as the resting phase. So during resting phase, we are having a one thing, that there is no stimulus, and another thing, we are having a term. And that term is what? That term is polarized state. And I am saying these terms with reference to the axon membrane. Okay, the plasma membrane, simple plasma membrane of the axon. Okay, from its point of view, from its state, I am saying that it is in polarized state. Next thing is the second stage. There is impulse, the nerve impulse is there. It means there must be a stimulus because of which there is impulse, and in this phase I will say that the membrane is in depolarized phase or the state. Okay, done, good. Then next stage is the restoration. First of all, there was no stimulus. Okay, the membrane was resting, the membrane was having polarized state. What does it means? We will get up to that point just in just a moment. Then what is the impulse state? Impulse stage is when there is a stimulus. Okay, when there is stimulus, the membrane is considered to be in depolarized state. And after that, just after that, third one is a restoration stage. In the restoration stage, there is again no stimulus. Sir, when there is no stimulus, then restoration phase is the resting phase also. Yeah, you can say that restoration phase is resting phase in terms that it is not carrying any impulse. But you will see in upcoming slides that how restoration phase are different from the resting phase. Okay, let me label it as repolarized state or repolarization state. Polarized, depolarized, and repolarized state. Okay, you can also coin some terms like polarization, depolarization, and ultimately repolarization. Okay, done? Understood? So, how the conduction of impulse take place? We are having three stages of axon membrane. Again, very important this thing. We are talking with reference to axonal membrane. So, axonal membrane is in polarized state, then depolarized state, and then repolarized state. So, how axon membrane is exhibiting these stages, that can be understood very well when we will be familiar with this term. And what is this term? This is membrane potential. What is the membrane potential? Let us discuss that thing. What is the basics? First of all, to make this concept very easy for you, let us get back to the basics of potential. You know, in physics, we are having a charge which is considered as q. Okay, and whenever we will have a charge, there will be an electric field associated with it, and then due to position of anything in the entire world, okay, in universe, we are having what? A potential energy associated with it because of its position. Okay, so since it is the case of charges, that's why we will say that it is electric potential. Understood? Okay, and when this charge will be in motion, then we can say that electric current is there. So in our body, a lot of electric current is there, while it is very minute in quantity. That's why we are so casual with it, and we are having electric potential as well. So this is the basics. Okay, hold on. This is just the basic. What is the potential doing here in the zoology? Okay, so we are having charge. Here we are not having electron moving like this. Okay, not like the current which is moving the wires, but we are having charged chemical species. Charged chemical species means ions. And here we are having a fact. What the fact is? Fact is that sodium ions and potassium ions, these two ions we are having in fact. So what is the fact? Sodium ion is the most abundant extracellular. Extracellular means outside the cell. So sodium ions are most abundant extracellular positive ions. Positive ions means cations. And similarly, the potassium ion are the most abundant intracellular cations. Intracellular cations. What does that it means? It means that in all the cells of our body, we are having sodium ions more outside the cell and potassium ion more inside the cell. And conversely, potassium ion less outside the cell and sodium ion less inside the cell. This is what? This is cells cytoplasm. This one. This is the cell membrane, general cell membrane, and this is the extracellular fluid, ECF. Got that? And if we consider this along the length of axon, then it will be more connecting to the concept. This is what? This is axolemma. And here we are having the cytoplasm of the axon, that is the exoplasm, and here we are having extracellular fluid. So considering about the axon length, we are having more sodium ions outside, less sodium ions inside, less potassium ion outside, more potassium ion inside. Question: sir, if both are monovalent cation, that that means both are carrying positive plus one charge only, then it is equal. No, how the potential is there? See, whenever we say that sodium ions are higher outside and potassium ions are lesser outside with reference to axonal membrane, here now we are talking about specifically axonal membrane, chloride ions and many negatively charged proteins are present in the exoplasm like neurotransmitters also. Okay, and they are impermeable, they are not going to diffuse in any direction. Okay, chloride ions will be there, the membrane is impermeable to them. So ultimately we can say that there is plus charge on the membrane, outside the membrane, and towards the interior of the axon or the exolemma it is negatively charged, outside is positively charged, relatively. Okay, so sodium ions are higher outside, potassium ions are higher inside, but along with that, chloride ions and negatively charged proteins are there permanent. They are not going to move across the membrane. So they are adding the negativity or negative potential in the exoplasm or towards the beneath underside of the exolemma. Okay. So here we are with this term, explaining this term, that is membrane potential. What is the potential? Due to positioning of any charge in a particular three-dimensional space, there is potential. And if we are discussing about this fact that sodium ion are most abundant extracellular cation, potassium ion are most abundant intracellular cation. Okay, you can see here how is it working along the axonal membrane. Okay, so that's why we can say that the exterior of the axon is positively charged, interior is negatively charged. Okay, credit goes to these two. Okay, and many other negative ions also. So one more thing, we are saying that membrane potential, no, membrane potential, we are not saying that sodium ion potential or potassium ion potential, we are saying membrane potential. What does that mean? Actually it is a little bit of misnomer because we are pertaining to membrane potential difference over here. Membrane potential difference, it means how much positivity is there outside, how much positive charge is there outside, and how much deficient the lower side of membrane is as compared to the outside. One side, second side. Okay, the second side is deficient in positive charge as compared to the first side. So there is potential difference. So this membrane potential difference is casually called as membrane potential. Okay, we are not again and again saying that membrane potential difference, just membrane potential. So what does it mean? It means the differential distribution, very important this thing, differential distribution of ions across a membrane. Here across axonal membrane is present. Okay, so this differential distribution of ions, it means sodium ion, it is not like sodium ions are higher outside, they are higher inside also. No, sodium ions are higher outside, they are less inside. Okay, so this is differential distribution. Similarly with potassium also it is applicable. So due to differential distribution of ions, there is a difference in the potential value of the charges, and that is known as the membrane potential. Understood? Now moving ahead, how this membrane potential is built and how it is getting changed during the nerve impulse conduction. To understand that thing, we have to understand the ion channels. Okay, let me revise you up to this point because it is very stepwise, but you have to be very careful while discussing this, while getting to know this. So we started from the nerve impulse, that is the information that is flowing from source to processing center, skin to brain, skin to spinal cord. Okay, and then we discussed about the generation, what is the generation of nerve impulse. Okay, what is the thing that we called as conduction? Conduction is the propagation. Okay, and transmission is transferring the nerve impulse from one neuron to another one. Then we discussed about the stages from axon membrane point of view. Axon membrane, when there is no stimulus, is polarized. When there is stimulus, it gets depolarized or excited state, we can say. Excited state, resting state, excited state, regaining or restoration state. So polarized, depolarized, and repolarized state. In order to understand this thing, that how axonal membrane is undergoing these changes, okay, we have to discuss this membrane potential. What were the basic? There is charge, there will be electric field, there will be potential, there will be motion, then there will be current. Okay, so not the electron, but we are having charged chemical species as ions, and mainly two ions. We are having the fact most abundant extracellular cation sodium and intracellular cation potassium. We discussed from axon point of view how we say that the exterior of exolemma is positively charged and interior is negatively charged because of chloride and negatively charged proteins. And now, what is the membrane potential? It is the membrane potential difference actually, because of the scarcity or the lack of some positive charge inside the axon. Okay, so this is known as the differential distribution of ions across the axonal membrane. How this differential distribution is maintained? Because see, we know that diffusion is the very miraculous process going on in our body all along the different tissues and different cells, because of which there is distribution of things along the concentration gradient. So if there are more sodium ion, they should diffuse inside, and if there are more potassium ion, they should move outside, and making the thing very clear. There should be equal charges. Sodium ion should be equal both sides, potassium ion should be equal both sides. But there is differential distribution. How it is maintained and then disturbed to form the nerve impulse, that we are going to discuss here. Okay, ion channels. So we are going to discuss about the ion channels. Ion channels are transmembranal proteins. These are what? Transmembranal proteins, okay, which are having their ends towards the cytoplasmic sites, as well as the extracellular side. Okay, this is the transmembranal protein. This is the lipid bilayer. Okay, this is the lipid bilayer. This is the transmembranal protein. This one. Okay, so this transmembranal protein form a channel, or we can say form a tunnel also, through which some ions or some material can move from the extracellular fluid to the interior of the cell, that is the cytoplasm. So talking about the axonal membrane ion channels, we are having gated ion channels and leak ion channels. Gated ion channels and leak ion channels. Okay, what do we mean by gated ion channels? It means they can be open. Okay, they are not always open, they are having a gate. It means any change in the differential distribution will excite them to undergo opening. Okay, so they can be open, if there is any disturbance in the amount of charges. Okay, or if there is any disturbance from the point of view of chemicals, if a certain chemical is present, then they can be opened. Okay, if there is certain charge is present, they can be open. Okay, if ATP is present, they can be open. Okay, so they are having a gate. Okay, very obvious, gated ion channels. Okay, and leak ion channels means, which, as the name suggests, they are always open. They are always open, through which the ions are leaking from the extracellular fluid to cytoplasm, or in both way. Okay, so getting into each one of them one by one, we are having here gated ion channels. Gated ion channels. They can be open or closed. One thing, they are gated. Okay. Next, they are of two type. A voltage gated ion channels may be there, which are in abbreviation known as VGIC. And next is the ATP gated ion channels. ATP gated means if ATP is present, they will function, otherwise they will not function. Okay. Then chemical gated ion channels. Chemical gated, it means like acetylcholine neurotransmitter. If it is present, they will be opened, otherwise not. Okay. So we have to emphasize upon these two right now when talking about the conduction. This will be used in the transmission thing. Okay. So voltage gated ion channels are separate for sodium and potassium. And ATP gated ion channel is common to both. Means what? Means on the length of axon, we are having what? The gated ion channels. It means the sodium ion gated channels or potassium ion VGIC, we can say. And then we are having the leak channels. Like this. Okay. So sodium ion VGIC, this is sodium ion VGIC. Sodium ion VGIC, potassium ion VGIC, voltage gated ion channel. This is sodium ion leak channels. Okay, sodium ion and potassium ion leak ion channels, LIC. This is not that LIC. Okay, yeah, much obvious. We are having this very important protein channels here also, that is known as the sodium potassium pump. By utilizing ATP energy, it is capable of moving 3 sodium outside and 2 potassiums inside. Okay, fine. So how it will be, how they will work. Okay, first of all, there is no impulse. There is no impulse. So the membrane is permeable to sodium and potassium both. But here is one fact, that in the resting state, the membrane is more permeable to potassium ions. Why is it so? Because the number of potassium ion leak ion channels are very high as compared to sodium ion leak ion channels, and because of which more and more sodium is capable of moving out. Okay, and it is adding into the positivity on the membrane or in the extracellular fluid, we can say. Yeah, okay, it is adding the positivity, and that's why the interior of the axonal membrane becomes 70 millivolt less potential than outside, and it is known as the resting membrane potential. That gives us value of minus 70 millivolt. Okay, look at this again. See, in the resting state, the exterior of the membrane is positively charged, interior is negatively charged, and the membrane is highly permeable to potassium ion because the leak ion channels are very high in number. We can say permeability of potassium ion is high, and it is adding up to this positive charge. Okay, and the interior become less. How less of potential? 70 millivolt potential due to these movement. And that's why just minus is just the convention, sign convention. Okay, because it is deficient. How much deficient of membrane potential is there in the interior? 70. So resting membrane potential is minus 70 millivolt. Next, as we have this stimulus, sodium ion VGIC got open and causing rapid influx of sodium ions. But one thing more, it is very short-lived opening. Very short-lived opening. So depolarization is done. Depolarization is done because of this influx. Because of this influx. Rapid influx. Okay. And the membrane become negatively charged outside and positively charged inside. Okay. Sodium ion when move inside the membrane, no. Okay. Then the sodium ions become greater inside the cell, inside the exoplasm. Potassium ions were already very high in the exoplasm, so they add the positivity in the interior, and it gives the positive charge, overall positive charge in the interior. Okay. Question. Sir, just a moment ago you said that potassium ions are moving very easily across the membrane towards the extracellular fluid, and they are adding the positivity. That's why it is 70 millivolt less. Yes, I said, but here the rapid word is very important. Huh? Rapid. There is such a rapid influx of sodium ions that beside this fact that potassium ion is very easily moving outside the membrane, the positivity is increased inside the exoplasm. Okay, got that point? This rapidity is very important in order to understand this concept. So this is how it is done. So it is depolarization. Okay, so from minus 70 to minus 60, minus 50, minus 40, minus 30, minus 20, minus 10, 0, plus 10, plus 20, ultimately plus 30. So this membrane, this new differential distribution of ions that is present here right now in this membrane is known as the action potential. Action potential, and that is plus 30 millivolt. That is plus 30 millivolt. Okay, so this is how depolarization is done. Now question again. Sir, we are having this potassium ion VGIC also, no? So why it is not opening? Why it is not opening? Why only this sodium ion VGIC is opening? See, let me be very clear here. We have made this structure only one membrane. Okay, only one membrane. Let me put this arrow like this. Okay, let me write here. That's 70 millivolt. 70 millivolt. This is the ECF. This is the lipid bilayer or cell membrane, and here it is the lower side of the membrane or the exoplasm. Exoplasm. Okay. Now next we were discussing about this. Next question, that why VGIC, potassium and VGIC are not open. Okay. See. These channels are proteins, and these proteins are made up of polypeptide chains, which are made up of ultimately amino acid. Chapter biomolecule, you must have familiar with that. So the amino acids are the deciding factor that up to which extent the sensitivity will be there of entire protein towards that voltage. Okay, so potassium ion VGIC is made up of a different set of amino acids and protein as compared to the sodium ion VGIC, and that's why it is less sensitive. So it will take a very little time, but yes, it is considerable. Up to that, action potential will be generated. Okay, action potential will be generated. Sodium ions were greater outside. After opening of VGIC, influx took place. It means the sodium ion VGIC is allowing the movement along the concentration gradient. No, along the concentration gradient. Okay. Now potassium and VGIC couldn't open. Why? Because they are less sensitive. Okay, and that's why there is a delayed response. Up to that time there is already action potential generated. Action potential is already generated. Okay, now there is repolarization state where the potassium ion VGIC got open. When the potassium ion VGIC open up, at this stage sodium ions are higher, potassium ions are also higher. When this channel is opening and potassium ion is going outside, okay, then what will happen? It is showing delayed response, okay, and potassium ion is higher inside, it is moving outside. It means it is also what? Along the concentration gradient. This movement is also along the concentration gradient. So VGICs and LICs both are allowing movement along the concentration gradient. So sodium ions are now higher inside, lower outside. Potassium ions are moving outside, making the exterior positively charged. Okay, and due to it will convert into potassium ion lower. Okay, during this process is occurring, and now repolarization has done. Okay, so potassium ion is lesser and outside is positively charged, so I can write like this. Relatively there is negative charge inside the exoplasm. So can you compare this with the polarized state? Let me show you this thing. Yeah, see the first stage, outside is positively charged, inside is negatively charged. Okay, then we are having the second stage. Okay, outside is negatively charged and inside is positively charged. And again this third stage, repolarization, you can very easily compare this to the polarized state. So electrically we can say, electrically the polarized state is equal to the repolarized state. Okay, but actually it is not. Okay, on the basis of differential distribution of ions, if we look at the differential distribution of ions, the polarized state is not equal to repolarized state. Why? Because this positivity is not because of sodium but because of potassium ions. Okay, earlier you remember, see again this slide, it was the sodium ion which were moving inside. Okay, this sodium ion here, it was moving inside. It means they were higher outside. Okay, they were higher outside, but here potassium ion here is higher on outside. So polarized state is because of the positivity outside caused by sodium and potassium both, where sodium ion were higher outside. Okay, sodium ions higher outside, but in this case potassium ions higher outside. This is the difference why a repolarized state cannot be considered as the resting membrane potential. Okay, so it is restoring back or reverting back or regaining back its polarity. So the generation we discussed, generation is done with the help of receptors. Okay, and the opening of what? Opening of sodium ion channels, or opening of sodium ion channels, and their influx create impulse. So this is the generation of impulse. How conduction is done? By creation of local circuit, that we will discuss just a moment after. Local circuit, creation of local circuit on length of axon, and then ultimately transmission will be done with the help of neurotransmitters that will open sodium ion channels on postsynaptic membrane. On postsynaptic membrane, just hold on, you will understand this. Let me write over here, postsynaptic membrane. So generation, conduction and transmission we discuss. Now what are local circuits? See, earlier it was all positive, interior wall was all negative. Okay, so this was the first stage, that is the resting potential. What happened in the uh, action potential thing? So we can say that positive negative, negative, positive. So what happened? We can have this, only this segment. In this segment, reversal of polarity happened. Reversal of polarity, that is the depolarized state. Okay, depolarized state happens. All along the length, it is quite like resting potential. So this segment of axon which is undergoing the depolarized state is known as the local circuit. Is known as the local circuit. This local circuit is made here. In the next step, the sodium ion channels which are present here will be induced to get open because of the potential difference here. Okay, it will be induced to get open, and this will be this segment which is local circuit, it will undergo repolarization while new circuit will be formed that will undergo depolarization. Let me make over here plus 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 plus. This will be having the repolarization. Okay, and a new circuit will be formed over here which will be like this, which will be like this. Okay, are you getting? Okay, so after that local circuit, again there is positivity outside and negatively charged membrane towards the interior. Okay, so local circuits are made due to induced opening of the sodium ion VGIC, and simultaneously there is closure of the sodium ion VGIC over here. Here it is opening, here it is closing. Okay, so repolarization is done here in the next step, and depolarization will be happening over here in this third step. Okay, so local circuits are formed, and from the dendrite to cyton and ultimately to synaptic node, this impulse is being transferred in the form of local circuit, that is the conduction of nerve impulse. Okay, I hope you all understood this, and ultimately we are having a note point here that during the resting state, VGIC are closed, leak ion channels are pumping potassium outside, sodium inside, sodium ions are higher outside and potassium ion are higher inside. This ATPase pump is playing very important role in the resting state. Okay, when depolarization was done, and after that when repolarization was reverting back the condition towards the resting stage or the polarized state. Okay, during this switching back, this sodium ion potassium ion ATPase pump is having very important role. Why? Because it has to restore the ionic distribution, the original ionic distribution, that is sodium ion should be higher outside and potassium ion should be higher inside. It is doing that along with the leak ion channels, but resting membrane potential is also maintained because of this. Why? Because sodium ions will try their effort maximum to use the leak ion channels and to get into the exolemma. Okay, so they are making sure that it should not happen, like sodium ion should not be lesser outside, they should always be higher outside. So it is only possible in the case that the 3 sodium ions are being pumped outside and 2 potassium ions are being pumped inside. It is also making sure that the positivity should be higher outside, despite of that fact that there is a negatively charged protein and chloride ion impermeable in state inside the exolemma. Okay, inside the exoplasm. Okay, so this was the note point. Next is the transmission. Okay, this is the last topic in our today's discussion. So how the transmission is done, let me make this diagram. That action potential arrives where we are having the synaptic vesicles full of neurotransmitters. Okay, and this is the post synaptic membrane. This gap is known as the cleft, synaptic cleft. I am explaining here only the chemical synapse. Okay, in maybe some other day in detail we will discuss the types of synapse, chemical synapse, electrical synapse, what is the difference, what is the benefit, what where they are found. Okay, what is the relation of them with respect to evolution, and that phenomena. Main was the membrane potential. Now we are finalizing this topic by considering the transmission of nerve impulse. So action potential arrives at the synaptic terminal. It leads to opening of calcium ion channels and influx of calcium ions. These calcium ions are there performing what? Calcium ion mediated exocytosis. Exocytosis, the phenomena of letting the vesicles get out of the cell. Okay, so this movement occur outside. Okay, it will fuse with the pre-synaptic membrane. When it will fuse, it will give out the neurotransmitters in the synaptic cleft. That is the space between these two, presynaptic and postsynaptic membrane. This postsynaptic membrane is it may be the dendrite. Okay, it may be the cyton of another neuron, it may be the axon of another neuron. Okay, so here we are having the receptors. Okay, these receptors, considering these neurotransmitters as, for example, acetylcholine, these acetylcholine will bind to these receptors. That will lead to opening of sodium ion channels and ultimately sodium ion influx. And what it will lead to, that you know very well, generation of action potential in another neuron. So that's why that's how the transmission of nerve impulse take place. First of all, let me revise for you. First of all, action potential reaches as synaptic terminals. Second, calcium ion influx and calcium ion mediated exocytosis, release of neurotransmitters and binding to receptors. Binding to receptors on postsynaptic membranes, binding to receptors and ultimately sodium ion influx and action potential generated in the next neuron. Okay, so it was today's topic where we discussed about the nerve impulse. We discussed that nerve impulse is generated with the help of receptors, then it is conducted because of ion channels and transmitted. Ultimately, three stages of axonal membrane we discussed: polarized, depolarized, repolarized. Ultimately, membrane potential we discussed. How from axonal point of view this membrane potential works, outside positive, inside relatively negative, that we discussed. Differential distribution of ion, this very important term we discussed. What type of ion channels are there, and when they work, in which condition they work, we discussed. And ultimately how conduction, how propagation is done by the formation of local circuit. We discussed one note point of role of this sodium potassium ATPase pump, number one. Number two is the point of one term is there, let us discuss that also: hyperpolarization. Hyperpolarization, it means resting membrane potential was minus 70 millivolt. Okay, it was raised up to plus 30 millivolt. Okay, when it is getting back because of potassium ion, it touches the resting potential and it dips a little bit before coming to the resting potential. This area is known as the hyperpolarization. And why is it so? Okay, guess why is it so? Guess why is it so? Guess. Because somewhere here, yeah, delayed response, less sensitive, and it is kept open a little longer than sodium ion VGIC. Potassium ion voltage gated ion channels, they are open a little longer than the sodium ion VGIC, and that lead to generation of a more negative uh, potential in the axonal membrane, that it becomes minus 90 or around minus 90 millivolt, minus 90 millivolt. Okay, and then ultimately they will close, and when they will close, the restoration is done with the help of these ion leak ion channels and with the help of this sodium potassium ATPase. Okay, last point is the threshold potential, that optimum amount of minimum amount of voltage generated across the membrane, along the membrane, so as the sodium ion, enough amount of sodium ion influx is done, okay, in order to have action potential. That is the threshold potential. So it is the minimum value of disturbance, I will say, disturbance created on neuron or axon. Okay, minimum value of disturbance, or you can in terms of ions you can say that enough amount of sodium ions influx is done. Okay, that's why there is closure of the gap between the action potential and the resting potential. That is, if some sodium ion will go inside, it will be like around minus 55 to minus 60 millivolt. If this difference is made, okay, after that it is a vicious cycle. The sodium, more sodium ions are going inside, it will create much less difference, that will induce more VGIC to open. Sodium ions will go out, go towards the exolemma, towards the exoplasm. The difference will be much less, then it will induce more sodium ion VGIC to open, and ultimately the plus 30 millivolt will be generated. So we can say here that threshold potential is always less than the action potential. This thing is also there. Okay, so we are done with the today's discussion here. Okay, thank you. See you in the next lecture. Thank you so much.